All right, guys, welcome back. We're going to pick up where we left off last time. We're going to finish active transport, and then we're going to talk a little bit about, review a flow chart about how these things work. Okay. All right. So if we remember, active transport needs ATP, which is just another term for energy. Okay. And we're going to talk more about ATP when we talk about cell energetics. So ATP, also known as energy. Okay. All right. So when particles pass through the cell membrane, but go from area of low to high. All right. So this is the inverse, once again, of passive transport. Passive, like I said, we had the ball, top of the hill, rolls down. It's passive transport. Well, now we kind of have the inverse. Okay. We have the ball. And even if I were to let's say people are like, oh, just throw it up the hill. Well, throwing it still requires energy. If I roll it or throw it, get it up to here, it's going to require energy, and that is in the form of ATP. Okay, so we are no longer here with the passive; we are here with the active, which is going to require some form of energy. It usually, always requires a cell membrane protein of some kind. So here, this is a sodium-potassium pump. All right, we got these two things, sodium potassium, going against the concentration gradient. Got something coming in, something coming out, depending on which thing we're talking about. All right, so as we just mentioned, ion pumps, sodium potassium is the one we'll talk about most. All right, this literally helps you do this. Okay, so without it, we're not going to be able to do it. So what we're doing is we're moving sodium to an area of low concentration. So let's say we have 1 over here to an area of higher concentration. Okay. And same thing with potassium, which is just K plus. Okay. We got 1 K plus on this side, and I'm moving it. Oops. And I'm moving it to an area with more K plus. Once again, against the concentration gradient from low to high concentration. Okay. So here's a very in-depth example. So you have the outside of the cell, and then you have the inside of the cell. So as you see here, we're pumping Na plus sodium out of the cell here. And when it opens and it changes that configuration, potassium comes in, and that phosphate breaks off from the ATP to ADP. And we'll, like I said, talk more about that later. All right, and then it kicks in the potassium. All right, so once again, low for sodium, high for potassium. All right, so we're kicking it out from low to high. Okay, so here we are as high for sodium, and we got low K plus or potassium. So we're kicking in the sodium, bringing in sodium from low to high, and we're kicking out, we're putting in potassium from low to high, putting it into the cell, kicking out sodium from low to high. All right. We got endocytosis. This is where we're going to bring in substances into the cell. They're going to kind of quote unquote eat. All right. So two types of endocytosis. We have pinocytosis or phagocytosis. All right. Pino means liquids. Phagocytosis means solids. This is just kind of flashcard stuff, right? You're going to need to know pinocytosis is liquids and phagocytosis is solids. And then exocytosis. So endo means in, exo just like exit means out. So this time we are getting rid of things. Could be waste, could be, hormones that are made in the body that need to be transported to other places. All right, we have our vesicles that we learned about in the last unit. They're going to essentially come up, merge with the cell membrane, spit those bad boys out. And it's not, you don't have to worry about your cell growing too much because probably like right over here where we can't see, you got something coming into the cell and it's engulfing that. So the cell membrane more or less stays pretty much neutral or the same size that it was. Yep. So gets rid of waste or secretes substances wherever they need to go. All right, so we're going to do a flow chart with these terms. So we have cell transport in a flow chart. We have two types. Write them in or think about them. Good. Passive and active. Now, each of these have three parts to it. All right. So here we have diffusion. All right. This is just particles from an area of high. Well, these are all from high to low concentration. You have diffusion, you have osmosis, and then you have facilitated diffusion. All right, so osmosis, high to low, but it is only referring to water. And these are high to low, but they need a protein. Okay. Things like ions and things like that. Okay. So now we have active transport. So we have ion pumps. That would be the sodium potassium pump. Okay. Now I have endocytosis. And exocytosis. Oops. All right. So endocytosis, we have two. We have pino and phagocytosis. All right. Remember liquid, solids. All right. And exocytosis is just out of the cell. Could be secreting hormones, waste, other things like that. Okay. So this is our flow chart. We're going to do a more in-depth one in class. So you can definitely use this as a template, but make sure to bring all these notes in because we're going to make a more in-depth one that you can use to study for the test. All right, guys, that's it for cell transport. If you have any questions, of course, fill them out on the form or bring them into class. I'll see you all then.